All right. Welcome. So today we're going to be talking about expression QTLs, or quantitative trait loci, where the quantitative trait is in fact expression. So slides credit goes to Ben Raby, who gave a guest lecture about this a few years ago, and Youngjin Park, who's a member of my lab. So here we are. We basically talked about population genetics last Tuesday, and then disease associations. So basically looking at the basic variation in genetic, signals for each person, and then how these genetic signals correlate with phenotype or disease association over here. And then today we're talking about how they correlate with intermediate phenotypes. And then on Thursday, we're going to look at heritability and how that works across the whole genome. So last Friday, we had the review panels. So who has already completed their reviews and discussed them in the panel? Great. Who hasn't? Okay. Are you guys meeting later on? Awesome. Good. And then reviews are going to be incorporated, returned back to the individuals, the individual proposers, and basically each group will then be able to then bring back revised proposals, and their responses will be due next Thursday. So we'll get those to you as soon as possible. All right. So first of all, for eQTLs, why do we care about mapping regulatory variants? Then we're going to go through an example of mapping methylation quantitative trait loci in Alzheimer's disease. And then we're going to step back and establish conceptual frameworks for working with eQTLs. And then a set of methodologies for actually carrying out eQTL discovery. And then we're going to go through a series of insights on eQTL function in biology that can then guide you when you carry out your own eQTL analysis. Then we're going to talk about allele-specific analysis, which complements eQTLs very nicely by looking within an individual at differences between one allele and another allele within the same cell. And then lastly, how do we use these techniques to then infer mediating genes through which genetics is acting on disease? So let's start with motivations. So the first motivation for why genetic variation in non-coding regions is extremely important. And then this is exactly what eQTLs are going to be about. So eQTLs, just to define them, are going to be quantitative traits that are based on gene expression, and then loci that guide these quantitative traits. So this is kind of like carrying out a genome-wide association study. Genome-wide association studies we typically think of as binary traits, where you have a case control cohort with quantitative traits such as height. You're not looking at tall, short, you're looking at a continuum of a distribution. And then you're asking about regression rather than classification. Just how much is the prevalence of the T allele, for example, correlated with the height of a person or the expression of a gene. So a quantitative trait basically is height. An eQT quantitative trait is basically a quantitative trait that pertains to the expression of some gene, usually nearby. And then L basically refers to the genetic locus where all of this is happening. So when we talk about an eQTL, it's basically a SNP, which is usually affecting a non-coding regulatory element, which then impacts the expression of a gene nearby. Okay? Raise your hands if you're 100% with me on this one. Great. That's at least what eQTLs are. Any questions on the definition? Who's not super clear on the definition of eQTL? I can expand it. Okay. So then, why do we care about non-coding variants? Basically, first of all, if you look at, this classical paper from 1975, Mary Claire King and Wilson basically looked at the number of differences between human and chimp in a bunch of important, genes and pathways. And what they found was almost no amino acid differences between human and chimp, which was a little striking. They basically said, wait a minute, they're just using human genes. Maybe they're just using them differently. So then they had this major supposition that evolutionary changes in anatomy and way of life are more often based on changes in the mechanisms controlling the expression of genes than on sequence changes in the proteins. So that was the first time that sort of the non-coding genome became so prevalent in people's eyes. The second one is just by sequencing the human genome and, realizing that the vast majority of the genome, 98.5% of the genome, is in fact non-coding. And then by sequencing a bunch of individuals and asking where are the variants across the whole genome, and then seeing that the coding variants are a tiny, tiny fraction of this huge number of non-coding variants. There are 15 million non-coding variants from the 1,000 genome data, only 100,000 coding variants. So, and then the third part is basically asking, what about disease-associated variants? And if you look at monogenic and Mendelian disorders, you basically find that, almost 90% of them are in fact coding variants. These are the strong effect variants. But these, again, are tiny in comparison to the huge, huge number of genetic associations that have been found for complex traits. And now nearly 90% of those are, in fact, in non-coding regions. And we talked a little bit about that in the population genetics lecture when we talked about how strong effect variants are usually maintained in the population at lower frequency because they are often detrimental. So the more of a phenotypic effect, a negative phenotypic effect you have, the less likely you are to be allowed to rise to high frequency. Everybody with me in this dichotomy between Mendelian and complex traits and between sort of very oligogenic and highly protein coding enriched strong effect variants versus highly, highly polygenic and non-coding enriched complex trait variants? Who's 100% with me on this one? Raise your hand. Awesome. Good. All right. So that's sort of the second motivation. So basically the idea here is that we have some kind of genetic difference between these individuals that leads to a difference in the expression of a gene that ultimately leads to the difference in the expression of the function of that gene, and then ultimately leads to some kind of intermediate phenotype and ultimately disease. Okay? So basically what we're going to be tracing through eQTL studies is what is the impact of the genetic variant on expression, but you have to think of that in the context of ultimately the function of that gene. Okay? What we're effectively trying to do is bridge that gap between genetic variation and disease by profiling intermediate phenotypes. So, we're in this lecture, we're going to focus on gene expression in specific tissues, but then this is in the path of endophenotypes and through regulatory variation. Okay? So that we can ultimately say, here's how, this genetic variant influences heart rate, for example, by acting in muscle, specific enhancer element, changing expression of some gene, and so on. Okay? And then the challenge, of course, with QTL studies is that every time you start looking at these intermediate phenotypes, they are no longer always causal to disease. I'm drawing here a causal path, but in fact, many of these intermediate variables might actually vary as a consequence of the disease, or both disease and these intermediate variables might vary as a consequence of the environment. So we have to develop methods to understand whether this particular gene is actually mediating the effect of the genetic variant on the disease. Okay? And that's going to be the last chapter of this lecture. Everybody with me so far? Okay. So I mentioned the FTO example last time of basically dissecting a non-coding association. So basically figuring out how a genetic variant is acting on the expression of nearby genes and how those nearby genes are in fact acting on cellular phenotypes and ultimately organismal phenotypes, and how we could actually dissect the exact function of that variant with the upstream regulator that normally binds the causal variant, specific tissue where it acts, and so on and so forth. And basically at the root of that was effectively an eQTL study. So it basically said, hey, the eQTL targets are IRX3 and RX5. What does that mean? That means that this disease-associated locus, that is the strongest association with obesity, acts by changing not the function of the gene that it sits in. So this variant sits in the FTO locus, but the FTO gene itself is not affected at all. You can see here the FTO gene doesn't change in expression. Instead, exactly two genes change in expression, and they're sitting 500,000 nucleotides away and 1.2 million nucleotides away. And this is the basis of an eQTL study. You basically look at individuals that carry the non-risk allele in blue and their expression levels, individuals carrying the risk allele in red and their expression levels, and then you look for the genes nearby that change in expression, okay? So that basically means that this genetic variant, RS1421089, that underlies obesity, is in fact an eQTL. It's an expression quantitative trait locus for these two genes, RX3 and RX5. Who's 100% with me so far? Awesome, great. So basically, why do we care about eQTLs? Because we can then go and intervene. Basically, if we have a path of causality between a genetic variant, you know, and ultimately a disease phenotype, we can basically say, if this is causal, that means that if I go and manipulate these genes, I should be able to see a causal impact on the phenotype, okay? And here, the feedback that the organism has on this variation is extremely important. Why? Because of homeostasis. Basically, what biological systems do best is try to remain the same, fighting off the forces of nature that would otherwise, you know, dissolve them up, okay? And that's what homeostasis is. Basically, it's the act of remaining the same. And what homeostasis means is that if I have, in the context of disease, a genetic variant that perturbs something, then there's going to be feedback loops that are going to try to fight against that something. And when I have a genetic variant that perturbs expression, most of the time it will be benign. But when I have a genetic variant that impacts all the way to disease, that basically means that the feedback mechanisms of the body did not make up for that genetic difference. And that basically means that if I go and perturb that same pathway, that I'm more likely to actually see a phenotypic effect. Who's with me on that subtle concept here? Great. So that's why we care about eQTLs. And what basically eQTLs allows to do is effectively trace that path of causality from SNPs to genes to cellular phenotypes to organismal phenotypes. And again, in the case of FTO, what you can see is that if you go and edit that single SNP, you can basically see an effect on the expression of the gene that's very far away, IRX3 and IRX5, both of them changing expression twofold by changing that single nucleotide. So that establishes this causality. And then I can go back and edit that SNP backwards, so basically from the edited allele back to the normal allele, and then you see this restoration of the expression. That also means, if that path of causality is true, that if I edit the gene, I should have an effect on the cellular phenotype. And indeed, if you overexpress the gene, or if you knock down the gene in risk or protective individuals, respectively, then you can see a change in that cellular phenotype of thermogenesis, of basically burning calories through heat. So that basically establishes causality from the gene to the cellular function. Then what about from the SNP all the way to the cellular function? So, you know, if that path of causality is true, then I should be able to see that impacting the SNP and editing the SNP can in fact impact the cellular phenotypes. And indeed, if you change the SNP, you directly see a change at the cellular level. And if that path of causality is true, then that means that also if I change the gene, I can see an impact on the organismal phenotype. And indeed, that's what we saw when we knocked down that gene in mice. Okay? Everybody with me as to sort of why eQTLs are so important? Because they can pinpoint the gene, which then gives you a handle to manipulate everything else. It gives you a handle for therapeutic development, for you know, establishing causality, for studying what are the cellular phenotypes, what are the organismal phenotypes. Who's with me so far? Awesome. Great. So let's look at such an example. So how do we carry out an eQTL study or a meQTL study in this particular case from beginning to end? Okay. So the example that I'm going to start with is basically looking at post-mortem brain methylation. So basically these are 750 individuals from the religious order study and the memory and aging project, the ROS-MAP cohort that David Bennett assembled a couple of decades ago. And all these individuals donated their brain post-mortem. So we can now start studying how are genetic variants in those individuals influencing the methylation patterns of their brain, and how are those correlated with the phenotypic differences of these individuals. So what we're going to do is basically measure methylation level at 480,000 loci using a methylation array. across the 750 individuals, okay? About half of whom have Alzheimer's disease. So that allows us to now start understanding the relationship between genetic variation and epigenomic variation. And we're going to do that in the context of the chromatin state annotation of these regions. Here's what an eQTL looks like when you actually visualize it. Up here, you have the chromatin state annotations for this region of the genome. So you have the gene annotations at the top on the forward strand and the reverse strand in dark blue and light blue. And then for every gene, you basically have its activity in a bunch of non-brain tissues and its activity in brain tissues. And you can see how, most of these genes appear to be active in brain, okay? And now all of this is in a reference cohort. What we're going to do for an eQTL study or for a methylation QTL study in this particular case, an meQTL study, is basically ask, number one, what is the average methylation that I observe in different kinds of regions? And what we can see here is, for example, this becomes, redder, which means higher average methylation in these enhancer regions, and it becomes lower, which means, less methylation in these transcribed regions. So basically, there's some correlation there with the underlying annotation of the corresponding region, okay? But then from that average methylation, we're going to ask about the differences, about the deltas between individuals. Is a person more methylated or less methylated at that location? And what you can see here is the red and blue, where red means more methylation, blue means less methylation, at every position across the 700 people, okay? Then at the same cohort, what we're measuring is the genotype of these individuals, whether I have 0, 1, or 2 alternate alleles. And that's in, green, yellow, and red. And the allele frequency is shown here. And then what you're asking is, are the individuals that had, more Gs, for example, showing more of a difference in methylation here, okay? So what we're basically looking for is a correlation between this vector of methylation across individuals and this vector of genetics across individuals, okay? And because of our, limitation in power, and also because of our prior belief that genetic variants act locally to affect methylation locally, we're going to be looking in nearby regions for these effects. And here's some examples where basically you see these genetic variants here are very strongly correlated with the methylation levels here, basically giving us an example of an or a methyl QTL. Okay? Who's 100% with me so far? Awesome. So basically that's at the visual level. Basically, what are the data that we're looking at? Any questions so far? Yeah. This thing here, how do I know that that's strong? Yeah, so basically this is the correlation between this vector here and that vector here. Visually, you can't quite see it, but when you take these vectors and you lay them side by side, I'm going to show you some examples. If you basically build a scatter plot, you'll see that the more of one particular allele people have, the more methylation they have. Does that answer your question? Yeah. Any other questions? Yeah. Yes. Yes. So this is a longitudinal cohort. So the individuals were enrolled when they were very old, like 80 plus years of age, and then they continued aging. And, more of them would develop Alzheimer's eventually. So then the question is, at the age of death, how many had already symptoms of Alzheimer's and how many had pathology of Alzheimer's? So we're going to, eventually get to the Alzheimer's variable. Right now, we're just ignoring that variable. We're just looking at genetics versus methylation. Does that answer your question? Yes. Any other questions? Okay, great. So then what's the first thing you do? You get all these massive matrices. What are you going to do? So somebody gives you, all of these data. The first thing you're going to do is basically ask about principal components of variation. What are the major forces that are driving the overall measurements in your cohort? Okay. Again, as statisticians, that's the first thing you should sort of think. How do I normalize the data? What are the drivers of variation in the data? And the first thing you want to do is basically, do some singular value decomposition or SVD of these analysis. So basically, you look at the principal components that arise from these singular values, and then you ask how much of the variation in methylation do I explain based on the first principal component, or the second one, or the third one, and so on and so forth. Okay. So you learn these completely de novo by effectively taking your very, very large matrix that I'm showing in this slide, and then carrying out SVD on it. Okay? Who's 100% with me on this one? So perfect. Just SVD. And then you basically ask, how many singular values should I consider? So then you can include more and more and more of those values. And then you can ask, well, how much of the variability do I capture? And what you can see is that the first few, all the way to 7, for example, have a sharp increase in variability. So they capture a lot of the underlying variability. And then, these capture less and less and less. They never stop capturing more. That's one of the unfortunate problems of genomic data. You basically can go down that rabbit hole as far as you want. You'll never get to the end of it. But they capture a lot of it, and then it sort of, tapers off. So basically, we decided to study the first 7 singular values based on this inflection point. And then we basically ask, well, what do these singular values mean? We'd like to have some biological interpretation for what they mean. And we therefore correlated them with known or measured covariates, shown here in blue. So for example, we looked at Alzheimer's diagnosis. That didn't capture much. Postmortem interval, that didn't capture much either. The specific study, whether it came from the really short study or the membrane aging project, that didn't capture much either. Okay? So it's kind of weird. It basically says that, all these things that you would expect influence, methylation greatly, did not affect it. Yeah. This was this methylation matrix. Say it again? Exactly. So yeah, methylation probe and individual. And the position doesn't matter that much. So even if you shuffle the columns, it's still fine. It's just methylation mark and individual. Okay? Everybody with me so far? All right. So then we can basically ask, well, then what are they? And we can, we can keep going down the list. The number of years of education, or the cell type mixture proportion. Basically, we can estimate what fraction is neurons, what fraction is microglia and astrocytes, oligodendrocytes. The plate on which this was carried out. So basically there were, 96-fold plates to do the experiment, which plate was used. The bisulfite conversion ratio. So basically when you measure methylation, what you basically do is treat your DNA in a way that distinguishes CpG methylation from, not methylated regions. And then, just how much of that bisulfite treatment did you carry out can be quantified as the bisulfite conversion ratio. And then the age and the sex of the individuals. And then you can ask, first of all, how are these measured covariates correlate with each other? And you can find here that which study you were in correlated greatly with what, how many years of education you had, what plate you were in correlated greatly with the bisulfite conversion ratio, and, your age and your sex correlated because, again, women live longer. And then Alzheimer's diagnosis also correlated with age, which kind of makes sense because the older you get, the more Alzheimer's you're likely to get. But then when we ask, what about these singular values that we learned? How can I explain those? And what we can see is that singular value number 1, for example, which correlated with value number 3 and value number 5, all of them correlated with both plate and bisulfite conversion rate. Singular value number 2 correlated greatly with sex and to a lower degree age. Singular value number 3, again with plate and bisulfite conversion, same with singular value 5. And then singular value 4 with cell type mixture component. Okay? And that basically shows you why it's so important to correct for these covariates. Because otherwise, you're going to basically say, you know, any kind of genetic variant that's enriched in particular plate, I'm going to call it as meaningful, even though it will just be an artifact of that plate being more bisulfite converted, and so on and so forth. Everybody with me on sort of this singular value composition? Yeah. No, they were randomized, but that's a very big question. Basically, when you put your samples onto a plate, you probably shouldn't put all your cases in one plate and all your controls in another plate, because then you will have no idea whether, you know, the case control difference was simply due to the plate difference, and so on and so forth. So you're picking out on something very important, that, you know, we should distribute the samples uniformly on these plates to ensure that that's not a main driver of the difference we see, because then we won't be able to distinguish the two. Other questions? All right, so then we can start asking, well, let's now, after we do all this correction and we remove all these, you know, 7 singular values and all of these known covariates, we can basically ask, you know, what remains and where does the variation happen? So then for every CpG, we can ask what is its average methylation, and you can see here the distribution of the average methylation, and you can see that, you know, it's a bimodal distribution. Many CpGs are simply unmethylated, and many CpGs are simply methylated. And then there's, like, you know, fewer in between. So you're either highly methylated or lowly methylated. And then you can ask, well, how variable are you? And then, so that's the CpG methylation standard deviation. And what you can see is that the more variable you are, the more you're sort of sitting in the middle. Basically, if you're all the way to the one end or the other end, you're typically less variable. And if you're intermediately methylated, then you tend to be more variable. And you can also break this down by the chromatin state annotation of the corresponding regions using the reference annotation. So remember that all of that is being done in the context of a chromatin state annotation. So then what you're finding is that, for example, promoter regions are very highly unmethylated. Transcribed regions are very highly methylated. It's kind of cool, right? That basically this distribution that we had no idea how to interpret before, as soon as you bring in the chromatin state annotation, makes a lot of sense. Promoters are simply, you know, unmethylated. Transcribed regions are simply methylated. Everybody with me here? We can do the same thing with bivalent and repressed states. Basically find that, you know, quiescent states, for example, are highly methylated, that these that these bivalent states are very highly unmethylated. And then we also figure out who's actually in the middle. Basically, where are all these variable sites falling? And where they fall is actually enhancer regions. So these H3K27 acetylation and H3K4 monomethylation, but no H3K4 trimethylation, which is the mark of promoters. This is kind of cool, right? We can basically start teasing apart the quote-unquote function, or at least the functional class of each of these methylation probes. And then we can basically ask, well, how variable are they across the different chromatin states? And what we find is that enhancer regions are by far the most variable, promoter regions are by far the least variable, and then it's a continuum in between, okay? And then once we've understood that, we can basically start asking how much does genetic variation influence CpG methylation, okay? So on the x-axis, I'm basically plotting the genotype, whether you have, you know, 0, 1, or 2 alleles of a particular SNP, of the reference, of the alternate allele of a particular SNP. And then just what is the CpG methylation level? Both of them normalized. Okay. And what I'm showing here is effectively a Manhattan plot for every single one of these methyl QTL tests. Basically, for every methylation probe, I'm asking, how much did that methylation probe correlate with genotype? And I'm overlaying 50,000 Manhattan plots on top of each other. Okay. For any one methylation mark, this would be, only one dot because we only tested the SNPs nearby. But the height of this dot basically tells you just how significant things were. And what's really remarkable here is that up there it says p to the minus 300. Okay, these p-values are astronomical. 10 to the minus 8 was the genome-wide significance threshold. In this particular case, because we're carrying out more tests, the genome-wide significance threshold is at 10 to the minus 11. But that's still that tiny little red line all the way to the bottom. Okay, so that basically means that in so many different locations across the genome, if I know the genotype, I can pretty much predict the epigenome. Okay? Who's with me so far? Raise your hand. Any questions? So then we can basically ask, how does methylation QTL discovery vary with distance from the methylation probe and with cohort size? And basically what I'm showing you here is if we only have 25 individuals, 50 individuals, 100 individuals, or 575 individuals that had both genetics and methylation, then what is the distance from the methylQTL SNP to the methylQTL target in KB, and what is the effect size between 0, 0.2, 0.4, 0.6, 0.8, and 1. And what you can see is that there's this inverse correlation here, where the further I am from the CPG that I'm observing, the further the SNP is, the less of an effect that SNP has. You basically see that the strongest effect SNPs are sitting very, very close to the CPG. Everybody with me on just looking at this bottom right plot here? The other thing you notice is just how your power increases with more and more individuals. You can basically say, with 25 individuals, I only have power to detect methylQTLs that have very strong effects. 80% of the variability in the methyl probe is in fact attributable to the genetics of that nearby SNP. And so on and so forth. As I increase the size, then I can discover things at lower threshold, and so on and so forth. And this is continuing this way. Another way to plot this is to basically say, how many methylation QTLs did I discover as I increase the size of the individual? And you can see here that this scales almost linearly. That means that we're nowhere near saturation. The more and more people you measure, the more and more methyl QTLs you'll discover. Okay, everybody clear with the blue curve here? And then you can also ask, well, what is the median effect size that is detected with additional individuals? Okay, so what is the minimum effect size? In this particular case, you can see that with more individuals, the minimum effect size detectable is in fact decreasing according to this red line. And also the median methyl QTL is decreasing as well. So that basically means that I'm finding more and more SNPs that are methyl QTLs, and these have smaller and smaller effect size. Okay, so certainly I discover more of them, but these have weaker and weaker effects. Who's 100% with me so far? Awesome. All right. So the other thing to ask is how reproducible are these methyl QTLs that you're finding? And then the problem is that as I carry out my methyl QTL, oh yeah, sorry. Yeah. So basically the question is, I have, I'm methylated or unmethylated, basically 0, 1. And then the question is, how much of that variability do I explain from the genetics? So this is basically a saying, do I have because I'm bounded between 0 and 1, my effect size can't be 10-fold or 20-fold and so forth. It has to be bound within that. So then what you're asking here is, effectively, what fraction of that variation between 0 and 1 can I explain using my methylation QTL? And basically here, these guys are explaining 80% of more of that methylation value. Whereas down here, these guys are explaining only 5% or 10% of that methylation value. Does that answer your question? No. These are all real. It's just that you have the power to detect them. Because if the effect is only 10% and I have 2 people, I'm not going to be able to say anything. If I have 10 people, I'm still not going to be able to say anything. Because, there's going to be some random noise. And then with more and more people, what you're doing is that you're making that distribution tighter. Another way to think about it is, how many times will I observe that allele? If that allele is at 50% frequency, for example, then with 10 people, I'm going to find it 5 and 5 times. If that allele is at 30% frequency, I need more people to even detect the allele. And then once I have the allele, I'm going to have 2 groups of individuals that are going to have different average methylation levels. And if the more individuals I get, the tighter that standard deviation is for each group. And the more I will be able to say that there is indeed a difference between these 2 categories. So that's where, power comes in. So basically, what's happening here is not that I'm finding noise, is that I'm detecting things that are becoming detectable that would otherwise be indistinguishable from noise. But because I have hundreds of people, I can now detect them reliably. Yeah. We're going to talk about LD shortly, but what do you have in mind? Yeah. Basically, should you treat them as separate hypotheses is your question. Yeah. So we're going to talk about some of that. Yeah, exactly. So you have to think of SNPs on the same LD block as multiple shots on goal. That's one way to think about them. You basically have a haplotype that's associated, and then you're asking, is each of the SNPs catching the effect of that haplotype? And that haplotype comes with all kinds of baggage. In addition to the common variants that define that haplotype, you have rare variants within it that might actually be ultimately responsible. So the shots on goal way of thinking about them is effectively saying, yes, there's some statistical fluctuation that would make different SNPs more associated or less associated within the same haplotype based on the LD breaking patterns of the cohort that I'm looking at relative to the overall LD structure of the population that I'm sampling these individuals from. And therefore, there's some kind of random breaking points of these haplotypes within the individuals that I'm looking at. And that can cause a lack of agreement, basically, because the true association comes from the causal SNP, but every now and then, other SNPs are going to be, riding along with it. If the haplotype block was perfectly defined in all the individuals, then I would have exactly the same p-value of association for all of the SNPs. But because of the random breakpoint across the different individuals, I have variation between them. This variation could be purely fortuitous, but that variation in the breaking patterns can also allow me to distinguish the true effect differences between different SNPs on the same haplotype. In my view, these are 2 separate problems. Number 1, detecting the association. Number 2, fine mapping the association. Once you have detected it, you can then ask, are they truly statistically different from each other? And then go about fine mapping. You can do conditional analysis. You can do all kinds of other things. You can start imputing rare variants and, sequencing that region. But the simplest way to think about this at this level is really just shots on goal. I have many SNPs. If one of them hits, awesome. If one of them hits, chances are another 1 or 2 or more will hit, and that's fine. Basically, we'll make the simplifying assumption that the true signal is coming from the haplotype rather than each SNP in that haplotype. Other questions? Okay. So we basically have these curves, and the third curve that I'm going to show here, which is the most worrisome of them, is basically the misestimation of the effect size, okay? As I have only 25 individuals, I'm feeling, you know, super fancy dandy discovering all these SNPs, and I'm like, woohoo, I'm doing great. Look at their great effect size. But it turns out that I'm greatly, you know, misestimating this effect size, okay? And you can see by how much, by almost 0.5. There's no doubt these are real, but they might actually have very small effects. And that's partly winner's curse. If you sample in one population, your true effect size might be here, and then you're going to be randomly sampling higher or lower than that based on the particular sample that you're drawing from the underlying true distribution of the population. If I sample slightly higher, I'm more likely to pass whatever threshold I set. And therefore, if something passes that threshold, it's more likely to be oversampled than undersampled, which basically means that in any replication cohort, it's more likely to regress back to the mean or even go the opposite way. So that's winner's curse, basically. As soon as you win the first time around, you pass that threshold, chances are you slightly overestimated its effect. And the next time you're going to measure it, you're going to be slightly below that threshold. Who's with me on that? No? Okay. Let me go over it again. Fighter pilots in Top Gun, they were rewarded for doing great in a battle. And then what they found is that every time you rewarded a pilot, the next time in a dogfight, they would do worse. So then they stopped giving awards because they said, oh, it's clearly that the awards are basically making the pilots fight worse. But the way to think about it is that there's awesome pilots and there's okay pilots, or maybe awesome and slightly less awesome pilots. We're talking about Top Gun. And the awesome pilots will sometimes perform slightly better than their average and sometimes slightly worse than their average. When you're setting a threshold for awesome performance, the awesome people will be slightly better than their average. And then you resample that same person after they get the award. The next time, they're just going to be sampling around that average. The award had nothing to do with it. Are you with me now? Good. So that's winner's curse. So basically, and that's where the term even regression to the mean comes from. They would regress to the mean the next time around. So basically, there's a true underlying distribution. Different people sit at different places in this distribution. Every time you sample, you move around that distribution. The same way, every SNP in the genome has some effect on the nearby SNP. You're sampling randomly in your distribution, in your sample of individuals. You sample 700 individuals from a population of millions. You're going to be bouncing around that true distribution. If I set a threshold for finding things that are real or that pass a certain threshold, I'm more likely to capture things that in my sample were slightly overestimated. Are you 100% with me now? Everybody's 100% with me? There's another question, yeah. There was another hand? No? Okay. Yes, no? Of course. But basically, we don't have an ascertainment bias. What makes a winner's curse is that we set a threshold and things that pass it, you know, have the combined effect of being awesome to start with and performing slightly better than average, than their average, which is a pretty good average. So if you look here, pretty much anything that I sample, that I discover, quote unquote, at this level, I way overestimate its true effect. And when I sample more people, that effect dramatically decreases. Okay? All right. And then you basically have the genomic distance of the SNP to the CpG, which basically creates this very strong concentration. You can basically say what fraction of my meQTLs are within, you know, a different distance. And here you see that 90% of my meQTLs lie within 75 kb of the CpG that they control. And 50% of meQTLs lie within, you know, the first 10 kb of the CpG that they control. So that basically means that if I want to increase my power, I can just simply ignore all the faraway guys. And then I have many fewer hypotheses that I'm testing. In this particular case, we actually searched for, you know, 200 kb in this graph, but we went all the way out to 1 megabase. And therefore, we were just shooting our power in the foot because you know, most of these guys would just not be true methylation QTLs. But as you know, but again, all of these guys are actually genuinely real. It's just that they're decreasing my discovery power because I'm testing many, many more hypotheses. Okay, and we're going to come back to that through the lecture to basically explain the trade-off between the radius that you're going to be looking at and your power. Who's 100% with me so far? Yay! Who feels that they've learned stuff? Raise your hand. Awesome! Yay! Who likes Top Gun? Yay! I was just in San Diego yesterday. I just landed back on another red-eye flight, and every 5 minutes you get zoom, zoom. And the guys were there telling me, oh yeah, they were filming Top Gun 2 recently, and it turns out that all these flights were flying slower and lower. I'm like, oh, why? Like, how much can you film a plane that's actually going at full speed? You have 3 frames on your camera. So basically, the actual pilots were just like totally slowing it down for us when we see these scenes and we're like, whoa, that went by fast. That's their slow flying. You should check out their fast flying. Anyway, sorry for the Top Gun example here. All right. So remember how we talked about earlier, if I have 20 individuals, I might not even detect that SNP. So we basically ask, what is the minor allele frequency of every SNP? And then how do I discover it with different numbers of individuals? And what you can see here is that as I sample fewer and fewer individuals, I'm skewing the distribution dramatically towards high frequency SNPs, because otherwise I wouldn't even see them with 25 individuals. So with only 25 people, you see that the SNP distribution of the ones that are methyl QTLs is in fact very highly biased. And then as I sample more and more individuals, I'm basically able to discover more and more of these low frequency SNPs. And then the true distribution of SNP frequency is the gray one. And you can see that I'm not even close to that. So I need to sample many, many more individuals to basically be able to truly capture SNPs that are at lower frequencies. Yeah. You know the frequency in the population. So you basically just ask for all of the SNPs, how often are they found in the population, and that's your gray distribution. And then you ask for the subset that are methyl QTLs discovered at each number of individuals. What does that distribution look like? Any other questions? All right. And why are we doing all this? Because once we're able to predict methylation from genetics, we can then take the predicted methylation and start correlating it with disease. And if I can do that, I get my causality back. Okay? Let me go through the different rows here. So this is what the thing that we're trying to break up, G to D, from genetics to disease, GWAS, genome-wide association study. That's a very, very long path. There are thousands of genetic variants that all impact your disease in a detectable fashion at 74,000 individuals. And as you increase that number of individuals, there will be more and more and more and more. Okay? So the reason why this path is so long is because, we're going all the way from genotype all the way to disease. And that basically means that every genetic variant that influences the disease ever so slightly, will be captured there with sufficient numbers of individuals. But that also means that the genetic variant that I'm able to detect will end up having, very, very weak effects, because any one SNP just doesn't influence disease by that much. Otherwise, that SNP would be selected against in the population. The individuals that, had that deleterious SNP on the disease would be, simply selected against. Or if that SNP is really, really beneficial and no one gets that disease, then that SNP might rise to fixation, and then the other SNP will, be much more deleterious. Anyway, so that's this very, very long path. I need many, many individuals, and the effects I will discover are very, very small. And an MWAS on the third row is a methylome-wide association study. So instead of looking at the association between genetics and disease, I'm now asking, well, let's measure the correlation between every methyl probe and the disease. Okay? So the problem with this is that it's a bidirectional arrow, because I don't know if the disease causes the methylation change or if the methylation change causes the disease. Who's with me so far between the first and the third row? Great. To do the third row, however, I need fewer individuals. I can do this with, 800 people because this is a less polygenic phenotype, okay? A more oligogenic phenotype because there's only so many methylation probes and these already summarize information from many nearby SNPs. So basically, this is likely to be stronger effect. And meQTL similarly bridges that gap by basically looking at methylation and its relationship with genetics. And then that you can do with relatively fewer individuals because there's, again, only so many genetic variants that influences methylation of any one loci. So methylation QTL basically does that. Now, an IMWAS, or an imputed methylome-wide association study, basically looks at how genetics predicts methylation, uses the predicted methylation value, and then associates that imputed methylation value with disease. That can be applied to many, many more individuals because all these individuals are able to share their genotype. It's much easier to get somebody to donate their genotype, donate their brain, because they're usually not done using it if you ask even nicely. Like, hold on to that. So we can basically impute methylation for 74,000 people and then correlate that imputed methylation with disease. So number one, I have more power because I have more individuals. Number two, I get my unidirectionality back because this is the genetic component of methylation. It's not exactly methylation, but I would argue that it might be better than methylation because this is the causal part of methylation. Okay? That's the part that I can predict before the individual is born. When I see the genotype of that individual at like 8 months of gestation, I can just say, well, when your baby is born and grows up to be 92 years of age, this will be their methylation level. So, that level I can predict when you're born. I can tell you when you're 92, your brain's going to look like that. And that is causality. Yeah. It's a very, very cool question. So you can ask for each person, what would be the predicted methylation and how does observed methylation vary for that person? And now you can actually start asking, what are the transacting factors that might be influencing this? Can I carry now a new QTL, which is a delta QTL, that basically tells me what are the genetic variants that influence the deviation of the observed methylation from the expected methylation for that person, and so on and so forth. Yeah, absolutely. Yes. Any other questions? All right. So when we do that, we basically find that there are many, many more loci that are now statistically significantly associated with Alzheimer's than what I could find at the genetic level. Okay. So here, I'm basically showing you what the IMWAS does. And it basically finds all these red loci that are now genome-wide significant above this red line. Because I have many individuals, I have fewer hypotheses, and so on and so forth. Okay? And if you look at the same SNPs here that control these genetic, that control these CPGs, then you find that, only one or two of them were basically, slightly above the fray. But again, none of them reached genome-wide significance. Genome-wide significance, would be above the top of the graph. Yeah. It's not that it needed them, but it could use them. So another way to look at this column is that I need them here, but here I can get away with 800. I can get away with 800, or alternatively, I can go all the way up to 800 because, that's how many people I can actually measure methylation for directly. Here, yes, I could have gone away with it with 800, but I can dramatically increase my power because I have the G from the GWAS, which is across 74,000 people. So then I can take that same G and apply it to many more people. Any other questions? Okay. So let's take a 60-second break, and then we're going to come back with the conceptual frameworks and sort of look at the theory behind all that. Okay, so everybody stand up. This is a mandatory 60-second break. All right, welcome back. So let's now step back and start reviewing what we saw, and hopefully almost every part of that lecture will be a review, but now in a sort of larger conceptual framework. Okay, so how do we map regulatory variance, gene expression as a quantitative trait? So basically, first of all, what causes gene expression variation? So, you know, you basically have some genetic variation at the cis regulatory element where the transcription factor normally binds, and also at the trans level where the transcription factor itself might have some genetic variance. And then you recruit polymerase, and then you know, that leads into some kind of difference in log expression. You can then decouple that by basically asking, you know, what is specifically the cis-acting variation and what is the trans-acting variation. So you can basically say how much of that variation that I see in expression is in fact due to these individuals having a different binding side, a different binding motif on the TF side, or having a different, so a different protein motif that basically recognizes a different protein sequence that recognizes a different motif across the genome versus a different motif instance that no longer matches the motif of the TF. So there's two things that can vary. One, the TF might actually recognize a different sequence. Two, that sequence that the TF recognized might simply not be there anymore. And basically, you can sort of study both the trans genotype of distant loci and the cis genotype of cis loci to try to decouple the two. Okay, so you can actually think of gene expression just like height as a quantitative heritable trait. You can basically ask, you know, how many of the transcripts have what kind of level of expression across generations. Then ask just what is the overall heritability for each gene. You can see that the heritability on average is, you know, 0.3, which is pretty heritable. Again, given all the caveats we have in actually measuring it. And we're going to talk next lecture about how measurement noise can affect your estimate of heritability greatly. So again, you have genetic variants that can influence, you know, the specific motif that the TF recognizes. A genetic variant that can influence the nearby methylation level and therefore might cause that TF to no longer bind. That genetic variant might influence the splicing or the post-transcriptional degradation of your transcript. It might influence your histone modification marks and the environment. It might influence high C domains and these topologically associated domains by impacting CTCF and so forth. So these are all cis-acting variants. But then in addition, these genetic variants can influence the trans aspect, namely on the transcription factor that binds these QTLs. So with cis eQTLs, the variant resides in close proximity to the target gene location. With trans eQTLs, the variant resides very, very distantly. It could be anywhere in the genome. And then the mechanism is much less clear. Okay? And now let's basically talk about how do we actually go about discovering these QTLs. So then the basic idea is that, you know, you isolate cells, you isolate RNA or DNA, and you measure methylation or histone modifications or RNA expression. And then you basically have a bunch of measurements across a bunch of individuals. Okay, so you measure all of these genes across all of these people. That gives you your expression matrix, just like we saw earlier. At the same time, you have your genotype matrix. Basically, genotype each of the individuals, and then you simply count how many alternate alleles do I have. And what eQTL discovery is about is effectively asking what is the linear regression equation that relates my expression to my genotype. I basically have some baseline expression for every gene. I have some slope that basically tells me how that basic expression, the basic level of expression varies with genotype, and then some amount of noise surrounding it. Okay? Who's 100% with me on this? Yeah, question? No, basically, because we're talking about quantitative traits, this is not a binary value anymore, but it's a continuous value. And that trait can be height or gene expression or methylation or splicing and so on and so forth. Does that answer your question? All right. Any other questions? Good. So basically, you know, there's now a difference between what is the true expression, what is the actual measurement, how many of these are clean. There's a QC to be carried out on the genotyping side. Some of these genetic variants might just simply, be bogus. And then there's all kinds of covariates, okay? And that's extremely important. Basically, in this particular case, expression is only influenced by genotype. But in reality, expression is influenced by genotype and all kinds of environmental factors. And these environmental factors include your age, your gender, some population stratification, all kinds of technical covariates, as we saw. So basically, when we looked at, this methylation matrix, the first thing we did was take out these covariates, and those covariates were associated with age, with gender, with study, and so forth. Yeah. That's going to be the next slide. So then, after you do that, you basically ask just, where do I draw my significance threshold and just how many variants are there in a couple of slides. So, but basically, the way that you do that is that you express expression or methylation or any kind of intermediate phenotype as a function of both your genotype and your covariates. So then anything that can be explained away using your covariates, you don't need to attribute to a genotype-driven variation, which I think is your question. So, you basically do a lot of feature selection about minimum expression levels, about minimum population variation, about how many SNPs might actually be disrupting my probes directly, how many reads are simply not mappable on the genome, and so on and so forth. So, I'm skipping a lot, as you can tell. Yeah, so this is one of the effects that we talked about last Thursday in terms of population stratification, or actually no, last Tuesday. We basically said, hey, if you actually look at the first principal components of variation, not of your methylation matrix, not of your expression matrix, but of your genotype matrix, then that genotype matrix is actually greatly influenced by your ethnicity, the population that you belong in, the history of, your ancestry. And the first thing that, we do in any such association is basically first estimate how much of that is actually coming from the population, which you can include again, either as part of your regression or simply regress it out, remove it, and then, use the residual. So you then have a bunch of decisions to make. So what is the threshold of minor allele frequency that you're going to be considering? What kind of normalization procedure you're going to be using? How many genes are you going to be keeping? How many nucleotides are you going to be looking around? So as I mentioned earlier, if you look at 2 megabases, you're basically hurting your signal because you're testing many, many more hypotheses, but you're discovering things that might be acting from far away. If you look very close, you have more power, but you're going to be missing things that act from far away. And, with the minor allele frequency, similarly, if you look all the way down to 1%, you have many, many more variants at, lower frequencies. The number of variants increases dramatically. So that means that you're testing many, many more hypotheses. But if you only look at 10% frequency, then you might be simply missing true effects of lower allele frequency variants. And those lower allele frequency variants, they are more likely to have stronger effects, because if something has a very strong effect, it's not going to rise to high frequency. So the ones that have stronger effects are in fact going to be at the lower frequency side. Okay? So then let's now study how does each of these impact our discovery power. So basically we can ask how many genes were found to have an expression quantitative trait locus at some discovery threshold, at the same discovery threshold. And the first question is basically at FDR. So basically my false discovery rate, I can choose a threshold of 1% or 5%. That basically means that 1 out of 20 predictions that I make is likely going to be bogus. And here it means that out of 100 predictions that I make is going to be bogus. And the more bogus predictions I'm willing to tolerate, the more genes with eQTLs I'm going to find. Many of these will actually be real, but 5% will be wrong. And here, only 1% will be wrong, but I'm losing many, many more genes. So, there's a decision to be made there. The other decision is just how close do I look? Basically, if I look at 75,000 nucleotides or 5,000 nucleotides, then, you can see that as I'm including more and more nucleotides, my power is actually decreasing. I discover fewer and fewer genes with methylation QTL or with expression QTL. And then you can also ask about minor allele frequency. So basically at very rare frequencies or more common and more common as my threshold. Basically what you can see here is that, I'm actually losing a lot of true effects by making the minor allele frequency be more stringent by basically only looking at more common variants. One would think, oh, maybe I'm increasing my power, but the effect of losing true signals is in fact dominating, and I end up discovering fewer QTL. Everybody with me on these? Awesome. So basically, this is sort of all these decisions that you're making can influence, your power greatly. And in fact, as you look at your minor allele frequency, your statistical power, will decrease as you go down the math because you're testing more hypotheses. But your true signal will increase because these guys are more likely to have an effect. And then you can see that that factor dominates, and you can find, many, many more of those SNPs. So then you can, do all kinds of things to basically ask, how do I choose the number of principal components, the minor allele frequency, and, finding optimality in that direction, okay? And then there's this very cool tool in Bioconductor that Ben Raby and his group developed that basically allow you to search that space very effectively and basically, carry out eQTL analysis quite easily. So now after you've done this a bunch of times, you can then come back and say, well, what had the biggest effect? And then, how are all of these different factors influencing my discovery power? You can basically ask what is the effect of eQTL on eQTL prevalence and impact, of different SNP filters, of how many genes do I find, where are these eQTL positioned, and you can see here how this number of eQTLs peaks right around the transcription start site. How reproducible are they? Basically, if I go across different cohorts, do I find that the effect sizes or even the directionality of the effect is consistent? How does the eQTL vary across tissue? So basically here, we can leverage a huge resource that was recently generated, the Genotype Tissue Expression Project. This was conceived, maybe a decade ago to basically carry out mapping of 40+ tissues across 1,000+ people, basically study the tissue-specific effects of genetic variants and gene expression. And you can see here, that with more and more people, I discover more and more e-genes, or basically genes that have an eQTL, and that across different tissues, the sample size varies because, we can obtain brain, for example, from as many individuals. So we have fewer brain samples, and it basically means that we discover fewer eQTLs. That's not because brain has less of a genetic effect on gene expression. It's simply because we have fewer samples. So as the sample increases, the number of e-genes continues to increase. You can also ask, how similar is the expression of different genes across tissues? And you can see here that the tissues basically cluster largely in a small number of very large groups. You have blood down here, you have internal organs here, you have brain here, and then you have muscle and heart here, and then some other organs here. So, that sort of basically tells you how the expression patterns cluster. But what about the genetic effects? Well, the genetic effects are driven both by the expression patterns, where the genes will even be expressed in those tissues, but also by the transacting regulators that are different from tissue to tissue. For example, gene expression might be very similar between heart and muscle, but in fact, the transacting regulator might be very different, causing a difference in the QTL. And then we can ask, well, how common are eQTLs from tissue to tissue? And you can basically rank all your tissues here and there, and then ask how much sharing is there. And what we can see is that different regions of the brain result in very similar eQTLs. So basically, there's a lot of sharing within brain, and then, with some exceptions. There's a lot of sharing within these internal organs, and there's like largely a big difference between the two. And then if you ask us how many tissues is a particular QTL discovered in, you basically see that it's a bimodal distribution. Some eQTLs are extremely cell type specific and tissue specific. These are discovered in only one tissue. And then many other eQTLs are discovered in all tissues. But then, if an eQTL is discovered in, say, more than 10 tissues, then it's going to be discovered in all of the tissues. So that basically says that there's really, tissue specific and tissue invariant QTLs. Who's with me so far? Great. And you can ask, what are the downstream genes? Can I start annotating those? Are they co-regulated? Are there transacting QTLs? And basically start linking the regulator and where that regulator is encoded in the genome with the target genes and where they are encoded in the genome. And you can basically start, studying that systematically, just by figuring out all of these edges in the network where a particular transcription factor is controlling different loci. You can ask, are they enriched in disease? You can basically find that eQTLs are in fact, very frequently enabling you to discover GWAS hits more easily. So if something is already an eQTL, it is much more likely to be a GWAS gene for many cases. There are again some exceptions, but basically if you ask, how many GWAS variants can I predict? Then if I don't use eQTL information, I sort of saturate there. But if I use eQTL information, I can go a lot further. So that basically means that eQTLs can actually help you in the quest to discover true disease genes. And then all of that has been about variation between individuals. So basically, I look at individuals that have 2 copies of the alternate allele, 1 copy of the alternate allele, and no copies of the alternate allele. And then I ask, how does the expression of a particular gene vary across these individuals? This is basically asking for individual variation. But then individuals themselves vary in age, in sex, in disease, and so on and so forth. Another question to ask is, well, what is specifically the cis component of that expression variation? And what is the best way to measure that? Well, the best way is to actually go within a person where all of the other covariates are matched. And then ask, is the A allele of that person in a heterozygous location differently active from the G allele at that same location in the same cells of that same person? Does that make sense? So basically what we're going to be looking at is, instead of differences from person to person, we're going to be looking at differences within a person by basically asking, what are all of the transcripts, for example, that I get for the same gene from the A allele of that person or the G allele of that person. And of course, for homozygous individuals, the AAs and the GGs, I can't do anything. So I throw them away. And I'm only looking at the GAs. And I'm basically looking at the heterozygous individuals and then asking, is the A allele more highly expressed than the G allele for these individuals? Okay? So that's allele-specific expression. Raise your hand if you're with me on this one. Okay, raise your hand if you're not yet with me on this one. I'll keep going. Okay, awesome. So every person is diploid. So we have 1 copy from mom and 1 copy from dad. In this particular case, I'm asking, well, you get an A from both mom and dad. how expressed are you? You get a G from both mom and dad, how expressed are you? And then you get an A from mom and G from dad, or vice versa. And what is your expression level? So you're effectively asking for the difference in expression between the whole gamish of genes that you have in your cell, okay? Without distinguishing what is the genotype of those transcripts. But because we're detecting transcript abundance by sequencing, I can basically go in and find all of the places where this particular gene had a variant that basically told me, aha, this is mom's copy of this gene, and this is dad's copy of that gene. And if I find that the copy of that gene that has an A is consistently more expressed, that basically tells me that there is a cis-acting difference that is causing that difference in expression. And that cis-acting difference is controlling for everything else, because the trans-acting regulators, they are exactly the same in that cell of that person. Any specific questions that I can help clarify? Who's with me now? Raise your hand. Awesome. Who's still not with me now? Okay, good. All the people who have a hand are with me. Great. And then you can sort of do a combination of those. So basically notice that the QTL analysis is primarily favoring these extremes, the AA and the GG. And the GA is usually in the middle. The allele-specific analysis is only looking at the AGs. The AAs don't matter and the GGs don't matter. You can combine the two by basically creating this combined haplotype test that's basically testing specifically for, you know, the total read depth across genotypes and the difference between alleles for those, and the allelic imbalance between the two alleles of the same individual for the heterozygous individuals. And we had a paper just a few weeks ago basically showing how the allele-specific loci that basically showed differences in methylation between one allele and the other allele are in fact enriched in GWAS loci. So that basically suggests that there are perhaps strong acting cis variants in that location that can help explain the difference in disease. We're also able to define epi-alleles, which are nearby epigenomic signals that influence the activity of an allele. The last concept I want to leave you with is this mediation analysis. I alluded to it earlier, the concept of being able to impute methylation and then use that in many more individuals. But there are two ways to do that. The first way to do that is to basically, so how does this work, the imputation-based association? So you basically have a reference panel, say GTEx, where you go and learn the association between genotype and expression. And then you have an application panel where you basically have the genotypes of these individuals. And then for every individual, you basically take their genotype, you apply the eQTL model that you just learned, and you predict their expression level. And then you associate these expression levels with a trait, okay? Who's with me 100% on the A part of this graph? Awesome, great. That's exactly what I described as IM was and the equivalent of IT was, basically imputed transcriptome-wide association. The problem is that not everyone is willing to share all of the genotypes of their cohort. Basically, you know, we may have 74,000 individuals in the Alzheimer's cohort. That doesn't mean that when we go and knock on their door and say, excuse me, could you please give us all the genotypes? They're going to say yes. Most of the time, they're going to say no, because they're going to say, well, if you have the full genotypes of individuals, you can guess who was in my study. And you can start predicting who has Alzheimer's disease and who doesn't based on their presence in my study. So another way to do that is to basically ask, what is the linear correlation between every location and expression? And then what is the linear correlation between all of the genotypes in aggregate in my cohort and expression? And then carry out the association at the level of summary statistics. So all I have is the association of every SNP with the disease. And I can basically start combining multiple SNPs together in a linear equation that is driven by the genetics of my reference panel. That basically allows me to now predict if expression as inferred by this combination of three SNPs, if expression as inferred by the combination of these three SNPs was predictive of Alzheimer's, then I'm just going to compute it directly on the summary statistics of the association with Alzheimer's for each of the SNPs. Because this association is linear, and because the GWAS association is linear, I can just carry out a linear combination of my associations to basically predict new associations through expression, imputed, but without ever looking at the genotypes of these individuals. Who's with me on this one? Raise your hand. Awesome. Very cool. So there's the reference underneath. So Gusev et al. And I really encourage you to read this paper. So what we're going to do now is go to the. Oops. Yeah, it's a little insane. And I'm not even putting everything on my calendar. This is just a group events.